this video, we're going to look at methods for studying cells. This includes the structure of both the electron and the optical microscope, magnification calculations, and cell fractionation and ultracentrifugation. The microscope was an important scientific invention when it came to developing our understanding of the microscopic world of cells and their subcellular structures. There are a few kinds that are used today: optical microscopes and electron microscopes. These fall into two categories: transmission electron microscopes and scanning electron microscopes, and we'll have a look at these in more detail in a little bit. We're going to start with optical microscopes. These were first invented in the 1590s, and they are the microscope mostly used in schools today. So we're going to have a look at their parts. We're going to start with the eyepiece lens. This is the section that you look through in order to see your specimen. We have objective lenses. There are usually 3 of these, and they're on a rotating disc. Usually they have a magnification of ×4, ×10, and ×40. Next, we have the stage. This is where you will put your slide with your specimen or object on. Underneath that, we have a lamp or a mirror in order to reflect the light up through the specimen. And finally, we have some focusing wheels, both a coarse focus and a fine focus. In the 1930s, another type of microscope was invented. This was the electron microscope. Now, electron microscopes have both a higher resolution and a higher magnification than light microscopes, allowing us to see images in more detail. This means we can see more organelles inside a cell than with the light microscope. The electron microscopes use beams of electrons rather than beams of light, and beams of electrons have a shorter wavelength than light, and this provides us with the higher resolution. So there are 2 that you need to know about: the transmission and the scanning, and we're going to have a look at these in more detail. First, a little overview. So in general then, they both have a higher resolution and a higher magnification than an optical microscope, okay? So the higher resolution here being about 0.1 nanometers, and we'll come on to exactly what resolution is in a minute, okay? They both have to work in a vacuum. So when those beams of electrons are being fired, those electrons would bounce off particles of air, so therefore we need to have it in a vacuum. If it's in a vacuum, our specimen must be dead. Okay, using a beam of electrons means that the images are going to appear in black and white. Okay, so they are detected and put onto a screen. It's not like we use our eyes in the same way with an optical microscope. And false colour can be added to the images. So you might have seen a lot of electron micrographs before of brightly coloured objects. However, this is false colour, so colour that we have added after the event. So 2 images at the bottom of the 2 types of electrons that we're going to have a look at, and we're going to start with our transmission electron microscope. So in this case, we can see our blue lines representing our beams of electrons, and they get passed through a series of electromagnets through the specimen in this case and detected by a screen at the bottom. Okay, now our specimen needs to be really thin as those electrons are passing through it. Okay, this then gives us a 2D black and white image called a photomicrograph, which is obviously processed at the bottom by a computer. Now the staining process of our specimen, which would be about here, is really complicated, and our final image can contain artifacts, okay? So artifacts are things that are not actually present in the specimen, but have kind of been picked up possibly by the staining process or by the fact that when these electrons are passed through, okay, sections will appear dark if they're absorbing the electrons and brighter if they are not. Okay, so there can be some discrepancies leading to artifacts, and when we're looking at our final images, they may not be a kind of perfect representation. Next, we have our scanning electron microscope, so slightly different. In this case, our electrons are fired at a specimen rather than through it. Okay, so our specimen is often coated in metal, usually gold, and because the electrons are hitting our object at the bottom and kind of scattering, they're detected and picked up by a detector, and they provide us with a 3D image. Again, it's still going to be black and white, so we can still add false colour to it, and our scanning electron microscope has a slightly lower resolution than our transmission electron microscopes. Okay, so more like 20 nanometers as opposed to the 0.1 that we saw before. Okay, so there are 2. We have scanning electron microscopes and we have transmission electron microscopes, and they can be shortened to SEM and TEM. Okay, so SEM for scanning and TEM for transmission. Okay, so the two kinds. Your transmission, your electrons are going through your specimen, whereas with your scanning, they are bouncing off and creating that lovely 3D image. When it comes to your written answers, definitions are so important, especially in this magnification topic. So, magnification is how many times bigger the image is than the object that you are looking at. So it's not how many times zoomed in you are, okay? How many times bigger the image is than the object that you are looking at. So the object is that real specimen, and the image either on a computer screen, in a textbook, on an exam paper. Okay, and resolution is how far apart 2 points can be distinguished as separate. So those different wavelengths that we were talking about, so your wavelength of light versus that wavelength of the beam of electrons, okay, provides us with the different resolutions. So the wavelength of light is slightly longer than the wavelength of the beam of electrons, so therefore those electron microscopes give us better resolution. The next thing that you need to be able to do when it comes to the microscope is convert between different units. These are really important when it comes to measuring, and you'll need to make sure that you are using the appropriate units for the size of the image or the object. Now, you may have heard of meters. These are probably going to be too big for measuring your cells. Next, we have millimeters. Sometimes these are also too big for measuring cells. Then we have micrometers and nanometers. Okay, so these ones here you may be less familiar with. In order to convert between them, we need to appreciate their scales. Okay, so we've got their standard form versions over there, which some people like. You need to be able to convert between all of these. Now, they're actually times 1,000 each time, which makes it nice and handy. So let's have a look at some of these conversions. So depending on the question that's being asked, you may have an image that's been measured in millimeters and they might want the actual size in nanometers, or it might be the other way around. You might also have to calculate magnification, and we'll come on to those questions in a minute. We're just going to practice these unit conversions first. Now, I have a handy diagram that I like to draw when it comes to doing these conversions, and that's where we're going to start. Okay, now if we are going from millimeters to micrometers and then over to nanometers. Okay, I like to be able to draw this diagram. So I like to go with some arrows. Okay, and if we're going in this direction, it's always going to be times 1,000. Okay, if we're going back the other way, then it is going to be divide 1,000. Now, it's important to note, if you're going from millimeters all the way to nanometers, it's not times by 2,000, it's times by 1,000, times by 1,000 again. Okay, so let's have a look at number 1. We're converting millimeters into micrometers. Okay, so question 1, we are going 1 millimeter to micrometers, so we're timesing by 1,000. Okay, so we're doing 1 times 1,000, which gives us 1,000. Now, units are really, really important. So you need to make sure you know which way you're converting. So in this case, we're into micrometers, so we need to add our units. Next, we're going 5 micrometers into nanometers. Okay, so 5.5, we're going from micrometers to nanometers. So again, timesing by 1,000. So 5.5 times by 1,000. Okay, so we're going from here to here, and that gives us 5500. Make sure we know which unit we're going into. That is nanometers. Finally, question 3. We are going from micrometers to millimeters. So we're going the other way this time. So following this arrow at the bottom, micrometers to millimeters. So this time we are divide by 1,000. So 37 divide by 1,000. Okay, and that gives us 0.037. And remember, we need to make sure we know which units we are. So micrometers into millimeters, we need millimeters on the end. Okay, now, if you don't put your units on the end of your answers, then it's unlikely that you will get full marks for the question. Along with converting units, you may also be asked to calculate magnification. This includes actual size, image size, and magnification calculations, and there is an equation that you can use. Let's work an example using this amoeba diagram over here. Okay, so what is the magnification of this image if the actual size of the amoeba is 30 micrometers? So we need that equation. Okay, so we're going to go m equals I over a. Magnification, image size, actual size, sometimes also called object size. And this fits into a nice triangle to help us rearrange that equation called the I triangle. Okay, so we are timesing across the bottom and dividing that way. So we are calculating magnification, so we can use this equation at the top. We know the amoeba's actual size is 30 micrometers. Okay, so the A in the equation here equals 30 micrometers. We now need the image size, okay? And you get that from measuring the actual image in your exam paper, okay? You need to put your ruler, in this case the scale bar is the easiest thing to measure. So going from 0 to about 88. We always measure in millimeters because that conversion from by 1,000 is much more logical than if you add in centimeters, because that's a factor of 10. Okay, so we have 88 millimeters. Now these units are not the same, so you would need to convert. Now I suggest in this case converting millimeters into micrometers because it keeps everything as whole numbers. Okay, so 88 times 1,000 gives us 88,000. Okay, so now you can put these numbers into your equation. So m equals, and we are going to do I over a, so image 88,000 divided by 30, and in this case it is going to give us a magnification of times 2,933.3. Okay, so key things: checking our units in the right place, checking we've rearranged this equation properly, and checking that we have added the relevant unit at the end. Let's have a look at another example. In this question, we want to know what the actual size in micrometers is of the nucleolus if the image has been magnified 2,750 times. So the nucleolus is the pink section in the middle of your nucleus. So let's put a ruler up against it, as that's what we will need. Okay, so between 6 and 7, that gives us 10 millimeters to work with. So let's do our triangle so that we can work out what our equation is going to be. Okay, we're looking for actual size. That means we're looking for A, that is image divided by magnification. Okay, like this. Now, we need to know what the image size is. We get that from measuring. Okay, so that gives us 10 millimeters. It wants the answer, however, in micrometers. So at this stage, I would be inclined to convert my millimeters to micrometers. So if we think back to that diagram before, we are timesing by 1,000. Okay, so 10 times 1,000 equals 10,000. And we need to make sure our units are there so that we know what we're doing. Next, we can put in our magnification value, so 2,750. So we've got image divided by magnification. Okay, so A is going to equal 10,000 divided by 2,750. Okay, so our answer for A is 3.636, which we can round up to 3.64. Now the only thing missing is our units. Again, it wants the actual size in micrometers, so you must make sure that you put your unit on the end or your answer doesn't mean much. In order for scientists to be able to discover and study the structure of different organelles within inside the cell, they developed a process called cell fractionation and ultracentrifugation. Okay, this is the process by which your samples are spun at increasing speeds to separate out the different organelles from within the cells. So let's have a look at how this works. Okay, we start with this process of homogenization. Okay, so our tissues are cut up, they are put into a solution of cold, isotonic and buffered solution. okay, and they are cut up. They are then placed into a blender to be homogenized further until all of those tissues have been broken down and your cells have been opened up. Next, your solutions are decanted and put into a centrifuge. Okay, so they might be put into test tubes or into centrifuge tubes, and they are spun at increasing speed. So this is an example of a centrifuge. Okay, your tubes will be placed into the slots here, lid closed, and then you would set it to spin, and you would do this at increasing speeds each time. Okay, so that first spin, you would remove your tube and you would have a separated solution. Okay, this top part being the supernatant and the bottom part being a pellet or sediment. Okay, so that supernatant is kind of decanted into your next tube. This would be respun at an increasing speed. Okay, and then again, it would separate into supernatant and pellet or sediment. This again would be taken out, put into a separate tube, and again would be spun at an increasing speed, and it would be separated out into your supernatant and into your pellet. Now, after the first spin, those heaviest organelles will likely sink to the bottom and form the pellet. Okay, so those are going to be the nuclei and also probably any unhomogenized tissue. Okay, within that second spin, organelles like chloroplasts and mitochondria are likely to be in the pellet or the sediment. And after that third spin, the lightest organelles like your lysosomes and your ribosomes are likely to be in that pellet. Okay, so these can be taken out each time. They can be popped onto microscope slides and you can have a look at what is available to see. At the beginning, we talked about the solutions being cold, isotonic and buffered. So cold in order to slow down enzyme action. We don't want any metabolic processes happening while we're trying to look at our organelles in case they might get damaged by those processes. Needs to be isotonic. This ensures that the solutions have the same water potential as the tissues. Okay, so this prevents any net movement of water kind of into or out of the organelles by osmosis because we don't want our organelles shriveling or bursting. Okay, so if the water potential wasn't the same, then the organelles could get damaged. It's important not to say cells. Okay, so we've broken open all our cells at this point, but it's the organelles that we are looking at. We don't want them to shrivel or burst, and the solution needs to be buffered. Okay, so we need to maintain the pH of the solution. We don't want any damage to proteins or organelles due to a change in the pH. Now that we've looked at the process of cell fractionation, let's check our key words. We're going to start with homogenize. So this is the process by which we break apart those tissues. Okay, so we cut them up, we put them in the blender to allow those organelles out. And in the case of plant tissues, we'll be breaking through that cell wall. Next, homogenate. So this is the solution after that process of homogenation, which we want to filter before putting into the centrifuge. This ensures we've removed any whole cell debris, okay, that wasn't broken down in that homogenation process. Next, the centrifuge. So this is the piece of equipment that spins your sample. Okay, so that filtered homogenate we will put into the centrifuge to be spun. Next, the supernatant. So once you've spun your solution, okay, your tube, you'll have the top part and the bottom part. That liquid part at the top is called the supernatant. That will be respun. Next, we have pellet or sediment. So when your tube was separated out, supernatant at the top, the pellet at the bottom. And depending on how quickly you have spun your solution, your pellet will contain different organelles based on mass. Okay, so these words you will need when you are writing an answer about the cell fractionation process. So in this video, we have looked at the structure of the electron microscope and optical microscope. We have looked at converting units and magnification calculations, and we have looked at the process of cell fractionation. In the next video, we will look at the process of cell division.